ability to repair the damage caused by toxic insults and inflammation is critical to the survival of an organism. The inflammatory response to microbes and injured tissues not only serves to eliminate these dangers, but also sets into motion the process of repair. The process of tissue repair is close to the idea of immortality. Why may not man one day be immortal? For many cultures, mortality is one of the major qualities that separates humanity from the gods. While humans are born subjected to the will of nature and die, the gods of the ancients and the gods of today are usually characterized as immortal, immune to the darkness that awaits every man and woman. Naturally, the earliest storytellers and holy men dreamed of ways to achieve immortality as well. In mythologies around the world, humans who achieve immortality are often regarded as gods or as possessing godlike qualities. One of the earliest works of literature, the 22nd century BC Epic of Gilgamesh, focuses on a hero's quest for immortality. In some traditions, immortality was bestowed by the gods themselves. Other times, a normal human would unlock alchemical secrets hidden in natural materials that stop death in its tracks. According to the ancients, the secret of immortality could be found within the earth or on the moon. In the 16th century, the story of the Fountain of Youth became attached to the biography of the conquistador Juan Ponce de Leon, who was a Spanish conquistador and explorer who was most active in the Caribbean in the early part of the 16th century. His name is usually associated with the exploration of Puerto Rico and Florida, where, according to the popular legend, he searched for the legendary Fountain of Youth. In Greek mythology, there is a figure of Prometheus, a titan, who stole fire from heaven and gave fire to humanity. The punishment of Prometheus as a consequence of the theft was very hard. By order of Zeus, king of the Olympian gods, immortal Prometheus was arrested and was bound to a rock. And each day an eagle was sent to feed his liver. And Prometheus' liver grew back to be eaten again the next day. This was the whole idea of the torture devised by the gods. The most important question there is how Prometheus' liver grew back. Today, we'll be discussing tissue repair. And this topic includes several parts, and maybe we'll be even able to find the question, to find an answer regarding the repair process and answer the question how Prometheus' liver was growing back. So we'll start with tissue repair, and critical to the survival of an organism is the ability to repair the damage caused by toxic insults and inflammation. The inflammatory response to microbes and injured tissues serves to eliminate these dangers, but also sets into motion the process of repair. Repair, sometimes called healing, refers to the restoration of tissue architecture and function after an injury. It occurs by two types of reactions: regeneration of an injured tissue and scar formation by the deposition of connective tissue. Some tissues are able to replace the damaged cells and essentially return to a normal state. This process is known as regeneration. Regeneration occurs by proliferation of residual uninjured cells that retain the capacity to divide, and by replacement from tissue stem cells. It is the typical response to injury in the rapidly dividing epithelia of the skin and intestines, and some parenchymal organs, notably the liver. If the injured tissue are incapable of regeneration, or if the supporting structures of the tissue are severely damaged, repair occurs by the laying down of connective fibrous tissue, a process that results in scar formation. Although the fibrous scar cannot perform the function of lost parenchymal cells, it provides enough structural stability that the injured tissue is usually able to function. The term fibrosis is most often used to describe the extensive deposition of collagen that occurs in the lungs, liver, kidney, and other organs as a consequence of chronic inflammation, or in the myocardium after extensive ischemic necrosis, infarction. If fibrosis develops in a tissue space occupied by an inflammatory exudate, it is called organization, as in organizing pneumonia affecting the lung. After many common types of injury, both regeneration and scar formation contribute in varying degrees to the ultimate repair. Both processes involve the proliferation of various cells and close interactions between cells and the extracellular matrix. The next section, we will be discussing the principle of cellular proliferation, the roles of growth factors in the proliferation of different cell types involved in repair, and the roles of stem cells in tissue homeostasis. And we will conclude with a summary of some important properties of the extracellular matrix and how it is involved in repair. The regeneration of injured cells and tissues involves cell proliferation, which is driven by growth factors and is critically dependent on the integrity of the extracellular matrix. Before describing examples of repair by regeneration, we should discuss the general principles of cell proliferation and the functions of the extracellular matrix in the process. Several cell types proliferate during tissue repair. These include the remnants of the injured tissue which attempts to restore its normal structure, the vascular endothelial cells to create new vessels that provide the nutrients needed for the repair process, and also fibroblasts that are the source of the fibrous tissue that forms the scar to fill defects that cannot be corrected by regeneration. The proliferation of these cell types is driven by proteins called growth factors. The production of polypeptide growth factors and the ability of cells to divide in response to these factors are important determinants of the adequacy of the repair process. The normal size of cell population is determined by a balance among cell proliferation, cell death by apoptosis, and emergence of new differentiated cells from stem cells. The key processes in the proliferation of cells are DNA replication and mitosis. The sequence of events that control these two processes is known as the cell cycle. At this stage, it is sufficient to note that non-dividing cells are in cell cycle arrest in the G1 phase or have exited the cycle and are in the G0 phase. Growth factors stimulate cells to transition from G0 into the G1 phase and beyond into DNA synthesis, G2 and mitosis phases. Progression is regulated by cyclins whose activity is controlled by cyclin-dependent kinases. Once cells enter the S phase, their DNA is replicated and they progress through G2 and mitosis. The ability of tissues to repair themselves is critically influenced by the intrinsic proliferative capacity. On the basis of this criterion, the tissues of the body are divided into three groups. Labile tissues, continuously dividing tissues, stable tissues, permanent tissues. Continuously divided tissues we'll consider over the next couple of minutes. Cells of these tissues are continuously being lost and replaced by maturation from stem cells and by proliferation of mature cells. Labeled cells include hematopoietic cells in the bone marrow, shown on the left picture, as well as the majority of surface epithelia such as pseudostratified, stratified, cellated epithelium, upper respiratory tract, shown on the right picture, squamous surfaces of the skin, oral cavity, vagina and cervix, the cuboidal epithelia of the ducts, draining exocrine organs including salivary glands, pancreas, biliary tract, the columnar epithelium of the gastrointestinal tract, uterus and fallopian tubes, and the transitional epithelium of the urinary tract. These tissues can readily regenerate after injury as long as the pool of stem cells is preserved. The stable tissues. Cells of these tissues are quiescent and have only minimal replicate activity in the normal state. However, these cells are capable of proliferating in response to injury or loss of tissue mass. Stable cells constitute the parenchyma of most solid tissues such as liver, kidney and pancreas. They also include endothelial cells, fibroblasts, and smooth muscle cells. The proliferation of these cells is particularly important in wound healing. With the exception of liver, stable tissues have a limited capacity to regenerate after injury. The picture that you see on this slide shows the fibrogenesis via epithelial to mesenchymal transition in the liver. Different insults initiate inflammation and then cause hepatocyte stellate cells activation and hepatocyte and biliary cell damage, necrosis, and epithelial to mesenchymal transition. Except for the necrotic and damaged cells, the remaining normal liver cells can be divided into two groups. Normal and epithelial to mesenchymal transition like hepatocytes. Continuous insults will shift those epithelial to mesenchymal transition-like cells to complete epithelial to mesenchymal transition cells, and finally, myofibroblasts, the main producer of extracellular matrix, which may be one of the main causes of an early loss of regenerative capacity. A similar process also occurs in biliary cells. Hepatocyte stellate cells play an important role in secreting key cytokines, such as transforming growth factor beta and hedgehog, to affect the adjacent cells and promote epithelial to mesenchymal transition. Let's talk about permanent tissues. The cells of these tissues are considered to be terminally differentiated and non-proliferative in postnatal life. Most neurons, shown on the left picture, and cardiac muscle cells on the right picture belong to this category. Thus, injury to brain or heart is irreversible and results in a scar because neurons and cardiac myocytes cannot regenerate. Limited stem cell replication and differentiation occur in some areas of the adult brain. Nevertheless, whatever proliferative capacity may exist in this tissue, it is insufficient to produce tissue regeneration after injury. Skeletal muscle is usually classified as a permanent tissue, but satellite cells attached to the endomysial sheath provide some regenerative capacity for this tissue. In permanent tissues, repair is typically dominated by scar formation, with the exception of tissues composed primarily of non-dividing permanent cells. Most mature tissues contain variable proportions of free cell types: continuously dividing cells, quiescent cells that can return to the cell cycle, and cells that have lost replicative ability. In most dividing tissues, the mature cells are terminally differentiated and short-lived. As mature cells die, the tissue is replenished by the differentiation of cells generated from stem cells. Therefore, in these tissues, there is a homeostatic equilibrium between the replication, self-renewal, and differentiation of stem cells and the death of the mature, fully differentiated cells. Such relationship is particularly evident in the continuously dividing epithelium of the skin and the gastrointestinal tract, in which stem cells live near the basal layer of the epithelium and cells differentiate as they migrate to the upper layers of the epithelium before they die and are shed from this surface. Stem cells are characterized by two important properties: self-renewal capacity and asymmetric replication. Asymmetric replication means that when a stem cell divides into one progenitor cell, shown on the left, and one stem cell, self-renewal, shown on the right, enables giving rise to two stem cells with higher repopulation potential or two differentiated cells. Although the scientific literature is replete with description of various types of stem cells, fundamentally there are two kinds: embryonic stem cells, ES cells, and adult stem cells. Embryonic stem cells are the most undifferentiated stem cells. They are present in the inner cell mass of the blastocyte and have extensive cell renewal capacity. Totipotent or omnipotent stem cells are embryonic cells that are created within the first few rounds of cell division after fertilization. Since these cells can differentiate into all the cell types, as well as the extraembryonic cell types, which play roles in the development of the embryo but do not become part of the organism after birth, omnipotent stem cells can build up a complex multicellular organism. An example of an omnipotent cell is a zygote, which is formed when sperm fertilizes an egg. Pluripotent stem cells are the daughter cells of omnipotent cells from multiple cycles of cell division. You could call them descendants of omnipotent cells. These cells can differentiate into almost every cell type from any of the three germ layers: endoderm, mesoderm, and ectoderm. Multipotent stem cells can differentiate into a limited number of cell types, usually those that fall into a specific range of cell types. Such cell types share some similarities and some differences, making them diverse to an extent. An example of multipotent cell is the mesenchymal stem cell, which can differentiate into bone, muscle, fat, and cartilage cells. Oligopotent stem cells are even more restricted in terms of differentiation potential than multipotent cells. The number of cell types they can differentiate into is very small. An example is the hematopoietic stem cells. Hematopoietic stem cells come from the mesoderm, the second germ layer, and can only differentiate into the blood cells. Unipotent stem cells are only able to differentiate into one cell type. An example of unipotent cells is a muscle cell, as it only differentiates into other muscle cells. Adult stem cells, also known as tissue stem cells, are less undifferentiated than embryonic stem cells and are found among differentiated cells within an organ or tissue. Although, like embryonic stem cells, they also have self-renewal capacity, this property is much more limited. In addition, their lineage potential ability to give rise to specialized cells is restricted to some or all of the differentiated cells of the tissue or organ in which they are found. Whereas the normal function of embryonic stem cells is to give rise to all cells of the body, adult stem cells are involved in tissue homeostasis. They maintain the compartment size both in tissues with high turnover such as skin, bone marrow, and gut epithelium, and in those with low cell turnover such as heart and blood vessels. Although there is much interest in isolation and infusion of tissue stem cells for replenishment of specialized cells in organs such as the heart after a myocardial infarction and brain after a stroke, tissue stem cells are rare and very difficult to isolate to purity. Furthermore, they occur in specialized microenvironments within the organ called stem cell niches.